Question 19. The diagram below represents a common laboratory equipment. Identify the equipment is a light microscope. Name the parts labeled E and H. E is clip or clips and H is body tube. Question C. Identify the letters that represent the part used in changing the parts labeled D from one to the other. That is a revolving nose piece, that's I. Used to incline the equipment to an appropriate position, that is G, that is a H screw. Used to make the image clearer when observing a specimen during a higher power objective lens, C, that is fine adjustment knob. A specimen was magnified 800 times using the above instrument whose magnification at part A was. What is the magnification of the objective lens? That is how you do the calculation. Question 20. The diagram below shows a certain cell. Use it to answer the questions that follow. Give 2 reasons why the above cell is not a plant cell. Has no cell wall, or you can say has central nucleus, or has centrioles, lacks large central vacuole. Name the parts labeled A, C, E, and J. A is the Golgi apparatus. C is centriole. E is the rough endoplasmic reticulum. Cytoplasm. Identify the letters that represent the part that produces ribosomes is D, produces lysosomes is A, synthesizes and transports lipids and steroids is H. State the function of the part labeled B. B is the bound cell content. It allows selective movement of materials in and out of the cell. Question 21a. What is osmosis? It is a process by which water molecules move from where they are highly concentrated to where they are lowly concentrated. Or you can say it is a process by which water molecules move from hypotonic solution to a hypertonic medium across a semi-permeable membrane. Study the figure and answer the questions that follow. The semi-permeable membrane, sugar molecules and water molecules. Which solution is hypotonic? Solution A. In which direction will osmosis occur? Solution A to solution B. What does the semi-permeable membrane correspond to in an animal cell? Cell membrane. Distinguish between the following terms: Tiger pressure and wall pressure. Tiger pressure is an outward pressure exerted by subvacuole against the cell wall when a plant cell gains water by osmosis. That is when it is placed in hypotonic solution, basically distilled water. And wall pressure is inward pressure exerted by cell wall against subvacuole when a plant cell gains water by osmosis. That is inward pressure exerted by cell wall that is equal to opposite Tiger pressure. Question 22a. Describe 7 factors that affect the rate of diffusion. Concentration gradient. It's an increase in the concentration of molecules at one region results in a steeper concentration gradient, which in turn increases the rate of diffusion. Temperature. High temperature increases kinetic energy of molecules. That is, they move faster, resulting in an increase in rate of diffusion and vice versa. Size of molecules or ions. The smaller the size of molecules or ions, the faster their movement, hence higher rate of diffusion. Density. The denser the molecules or ion diffusing, the slower the rate of diffusion and vice versa. Medium. The medium through which diffusion occurs also affects diffusion of molecules of ions. For example, diffusion of molecules through gas and liquid media is faster than those through a solid medium. Thickness of membrane. Rate of diffusion is faster when the distance is small, or when the distance is the rate of diffusion is faster when the distance is small. Or you can say the rate of diffusion is faster when there is a thin surface. Surface area to volume ratio. The larger the surface area to volume ratio, the faster the rate of diffusion. For example, in small organisms such as amoeba, the surface area to volume ratio is greater and faster diffusion than in larger organisms. B. State the role of osmosis in living organisms. Supporting plants, especially herbaceous plants. Opening and closing stomata. Absorption of water from the alimentary canal in mammals. Reabsorption of water in kidney tubes. Movement of water into cells from the surrounding tissue fluid and also from cell to cell. Did you understand? Kindly let me know in the comment section below. Also let me know what video I can look at tomorrow. Thanks a lot for subscribing and listening to me. God bless you.